This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
Good afternoon, everyone. I'm Mary Camario, professor of the Graduate School in Architecture and chair of the Hitchcock Professorship Committee. We are pleased, along with the Graduate Council, to present Carol Greider, this year's speaker in the Charles M. and Martha Hitchcock Lecture Series. As a condition of this bequest, we are obligated and happy to tell you how the endowment came to UC Berkeley. It is a story that exemplifies, in many ways, the campus link to the history of California and to the Bay Area. Dr. Charles Hitchcock, a physician for the Army, came to San Francisco during the gold rush, where he opened a thriving private practice. In 1885, Charles established a professorship here at Berkeley as an expression of his long-held interest in education. His daughter, Lily Hitchcock Coit, still treasured in San Francisco for her colorful personality as well as her generosity, greatly expanded her father's original gift to establish a professorship at UC Berkeley, making it possible for us to present a series of lectures. The Hitchcock Fund has become one of the most cherished endowments of the University of California, recognizing the highest distinction of scholarly thought and achievement. Thank you, Lily and Charles, and now a few words about Carol Greider. Carol Greider is internationally renowned for her important contributions to the field of molecular biology. She was awarded the 2009 Nobel Prize for Physiology of Medicine, along with Elizabeth Blackburn and Jack Szostak, for their discovery that genetic sequences known as telomeres are protected from progressive shortening by the enzyme telomerase. Greider discovered telomerase in 1984 while working with Elizabeth Blackburn at the University of California, Berkeley. Telomeres are essential to chromosome maintenance and stability. In the absence of telomerase, telomeres shorten progressively as cells divide, eventually leading to cell death or cellular senescence. Greider's breakthrough discovery of telomerase enzyme that allowed a mechanistic understanding of cellular reproduction, opening new avenues of research into cancer and age-related diseases. Greider received her BA in biology from the University of California, Santa Barbara in 1983, her PhD in molecular biology at the University of California, Berkeley in 1987. She was a Cold Spring Harbor Fellow and later held a faculty position at the Cold Spring Harbor Laboratory in Long Island, New York. In 1997, she joined the faculty of the Department of Molecular Biology and Genetics at Johns Hopkins University, where she is currently the Daniel Nathans Professor and Director of Molecular Biology and Genetics, as well as Professor of Oncology in the Johns Hopkins University School of Medicine. Greider also directs her own research laboratory at the university, which conducts research into telomere function and structure. In recognition of her groundbreaking research, Greider has been named a fellow by the American Academy of Microbiology, the American Association for the Advancement of Science, the American Academy of Arts and Sciences, and is a member of the National Academy of Sciences and the Institute of Medicine. In addition to her Nobel Prize for Physiology of Medicine, she has received a number of other awards in the last 10 years, including the Perlmeister Greengard Prize, the Paul Ehrlich Ludwig Darmstadter Prize, the Catherine Birkin Judd Award, the Richard Lounsbury Award, the Dixon Prize in Medicine, the Wiley Prize in Biomedical Sciences, and the Louisa Gross Horowitz Prize, an amazing list. Greider has also published over 100 articles and book chapters. Please join me in welcoming Carol Greider back to Berkeley.
Well, it's really a pleasure to be here. I've been looking forward to this lecture series for a number of months now. It's always a pleasure to come back here to Berkeley and to be here in the I House. Not only did I get my PhD here at Berkeley, but both my mother and my father received their PhDs here at Berkeley. So it really is like a homecoming. So thank you very much for this invitation. So what I would like to do today is to give you a sense of the background of why we thought telomeres and telomerase were interesting, and take you on a little bit of a journey from the initial discovery of the function of telomeres through how now we understand they play a role in age-related disease. So the theme of this lecture is going to be that curiosity-driven research can establish fundamental knowledge that fuels practical applications. So we'll start at the beginning, or perhaps we'll start at the end, because telomeres are at both the beginning and the ends of chromosomes. So if you have within a cell the nucleus, and in the nucleus are all of the chromosomes, and this is a blow-up of one of those chromosomes. And so most of what is studied in the field of molecular biology occurs along the length of the chromosomes, where all of the genes and the control elements lie. But we'll be focusing today on this region at the end, which is called the telomere. Telomeres provide two really important functions for the cell. First of all, they have to protect the chromosome ends. They protect the chromosome ends from anything that would chew away a nuclease that may chew away from the end. They protect the ends from recombination with other ends, and they protect the ends from fusion with other ends. So this is a really essential function for chromosome maintenance. The other thing that telomeres have to do is to allow the maintenance of chromosome length. And we'll be going through why that is very important throughout most of the rest of the talk. So telomeres were first functionally defined by H.J. Muller and by Barbara McClintock back in the late 30s and early 40s. So Muller, working in Drosophila, showed that telomeres were essential to protect chromosome ends, and McClintock was working in the plant maize or corn, and also came to the same conclusion that the telomeres were necessary for protecting the chromosome ends. So this was a very early discovery, and it wasn't possible to really understand in molecular detail what a telomere was or how it carried out its function until it was characterized at the very molecular level. And so that occurred in the 1970s with this organism here, Tetrahymena thermophila. So Tetrahymena is a single-celled ciliated protozoum. If you went to the creek, you'd be able to take some pond water and see ciliates swimming around. And the tetrahymena is very unique because a single cell contains 40,000 chromosomes. So this was the organism of choice to try and identify what is the molecular basis for the function of these telomeres. And so in 1978, Elizabeth Blackburn, working together with Joe Gall, were able to define the molecular sequence, the DNA sequence that's at the very end of the chromosome. And what they found is that there was a very simple repeated DNA sequence. So the DNA has normally 4 different building blocks, T, G, C, and A. And in this case, it was simple repeats of 2 T's followed by 4 G's, and then hundreds of those repeats on the ends of the chromosomes. So this was a very important initial finding because it then allowed additional experiments to be carried out to understand the function of telomeres. So here's Joe Gall and Liz Blackburn. I'm going to introduce a number of the people along the way, because one of the themes in science is, of course, it's not just one person making a discovery, but many people building on what others have done. So I'll try and give you a flavor of some of the people that have done the experiments that I'm talking about as I go along. So the first identification of the DNA sequences of telomeres, as I said, was in this ciliated protozoan Tetrahymena, and the telomere sequence was identified as T2G4, repeats of this sequence. And then subsequent work by a number of other laboratories was able to show that very similar tandemly repeated simple GT-rich sequences were present in a variety of other organisms. So for instance, in fungi, there's a model organism, Saccharomyces cerevisiae, and here the sequence was T followed by either 1, 2, or 3 G residues. And then I'll also be talking about the human telomeric sequence, and in all vertebrates, including humans, the sequence is TTA-GGG, repeated many times. So this theme, it turns out, repeats itself throughout various organisms. And this is one thing that's very nice about working on something that is very fundamental to biology, and that is that the same themes occur throughout many different organisms. So the telomeric DNA contains these tandem repeats of simple sequences. And of course, most of the DNA in a cell is double-stranded. So you have both the TG-rich sequence as well as a CA-rich sequence, although there is a region at the very end of the chromosome in which it's single-stranded. But in order to carry out this function of telomeres, you require not just the DNA, but of course there have to be proteins that bind to that DNA that actually carry out the function of protecting the chromosome. And in mammalian cells, this is a complex of proteins called the sheltering complex, which protect the chromosome ends. And so there are proteins that bind along the length of the double strand of the DNA, proteins that bind to those proteins, and then proteins that also protect the single strand region. And it's together, this complex of the proteins in the DNA that carry out the telomere function that we'll be discussing today. So it turns out that it was first hypothesized by Jim Watson and by others that if you look at the mechanism by which chromosomes are copied every time a cell has to divide, that it was predicted that the chromosomes should shorten from their ends every time the cell divides. And this is because of the molecular mechanism that replicates the entire length of the chromosome. So what one would predict is that every time a cell divides, the telomeres should shorten from their ends. And so you would have a loss of these telomeric repeats. However, it turns out that the way most organisms that have these linear chromosomes overcome this problem in replication is through this enzyme telomerase. And that's what we were able to show from Tetrahymena. So you may have some telomere shortening here, but then the telomerase will add these repeats back onto the ends of chromosomes so that chromosomes aren't a unique length. Rather, there's an equilibrium. There's always some shortening, and then some lengthening, and some shortening, and some lengthening. And that way, the average length of the average telomere is maintained over time. So this is a rather Baroque mechanism that doesn't really avoid the shortening problem, but rather overcomes it. So we've been interested in this telomerase enzyme for a number of years, and of course we don't usually look at it as this kind of a little cartoon. We're interested a little bit more in the details of the mechanism, but I won't really have a chance to get into many of those details here today, but I'll just show you some of the components that go into this telomerase so that I can tell you a little bit about how we study its role. So the telomerase is a really remarkable enzyme. It actually contains protein components as well as an essential RNA component. So this is unusual for an enzyme. So this stick figure here is meant to represent the RNA component of telomerase. And the RNA component of telomerase actually has a region which can base pair with the telomere to be able to add these telomeric repeats onto the ends of the chromosome. And I'm introducing this not so much today to tell you about how telomerase does this, which is really a fascinating question, but rather to introduce you to the components of telomerase that I'll be discussing throughout the talk. So this protein here that carries out telomerase function, we call TERT for telomerase reverse transcriptase. And so this carries out the catalytic function of telomerase. And this RNA component we imaginatively call telomerase RNA, abbreviated TR. So you'll be hearing about these components throughout the rest of the talk. So having discovered that the way that telomeres are maintained, rather than having them shorten every time the cells divide, rather to be maintained by telomerase, we were very curious to know what would happen if you don't have telomerase. What would be the consequence of not being able to elongate the telomeres? And so that led us and a number of other labs to two really fundamental consequences of telomerase. So the question is, why does telomerase matter? And it turns out that it's required for all cells that have to divide many times. So I'll be telling you a little bit of a story where telomerase is required for the growth of cancer cells. So if you imagine that you have a particular set of cells here, one of those cells undergoes some kind of a genetic change which allows it to divide more times than it normally should. So that's this red cell here. And that red cell then will go and divide many more times relative to the other cells around it. And it turns out, in order for that cell to continue dividing and form that tumor, it has to solve this telomere problem. That is, the problem with the telomere shortening every time the cells divide. So these cancer cells need to have telomerase in order to maintain their telomeres to go on and continue dividing. So I'll tell you a little bit about that. Telomerase is also essential for a number of other cells in our body, which have to also divide many times. And these would be stem cells or tissue-specific stem cells, where you have a cell that makes a copy of itself, but then also has to make what are called pluripotent stem cells. These are cells that will go on and make the final tissue. So this, again, are cells that have to divide many times in order to replenish the other cells in the body. So these are the two areas that telomerase clearly plays an important role. And we'll be talking toward the end of the talk about how this role here plays a role in age-related degenerative disease. So we were very curious about the role of telomerase and what might happen if you didn't have telomerase. So we set out to generate a mouse which doesn't have this activity. So instead of being able to maintain the telomeres by adding repeats onto the end, we generated a knockout, so it had no telomerase at all. And then we wanted to study what happens to that mouse when the telomeres get to be short. So the way that we generated this mouse that doesn't have telomerase was we had identified, as I told you, the telomerase RNA. That's abbreviated here TR. The M stands for the mouse, so it's the mouse version of the telomerase RNA. And we generated mice because mice are diploid. Of course, they have two copies of chromosomes, one from their mom and one from their dad. So we generated these mice, which are heterozygous, in which they have one normal copy of the telomerase RNA, and then the other copy is just missing the telomerase RNA. So it's minus. So it has one copy instead of two copies. When you then take two such mice and you breed them together, as Mendel told us from genetics, what you would get by breeding these two mice together is you get mice that have two copies of the telomerase RNA, mice that are heterozygotes, or have, like their parents, one normal copy and one missing copy. Or you get the mice that are completely null for telomerase and have no enzyme whatsoever. So we generated these mice, and remarkably, the mice were perfectly fine. There was nothing wrong with them at all. And so we were curious what might happen over time with these mice. So we designated these mice as the G1, that is, the first generation that lack telomerase. So these G1 mice were then bred to each other. And of course, you breed the G1 mice. So these are null. You get the G2 knockout mice. You breed those mice together. You get G3, et cetera, G4, G5, and G6. And I'll be showing you that in these G6 animals, we can't get a later generation because one of the things that happens is these mice become infertile. There are a number of other consequences of the loss of telomerase, but we don't get future generations. So as these mice are breeding, what we found is that the telomeres with each generation of mouse were getting shorter and shorter. So the cartoon version of this is shown here. We breed the G1 mice and we get the G2. We take these two G2 mice, and now the G3 mice have shorter telomeres than their parents did. And then you breed the G3 mice to each other, and the G4 mice have even shorter telomeres. So with progressive generations, there was progressive telomere shortening. And the way that we actually look at this in the laboratory is a very nice visual assay that was developed by Peter Lansdorp. And what this is a way to look at all of the chromosomes in the mice. So these are metaphase spreads. So you take the cells when they're about to divide, and you can visualize the chromosomes, which are shown here in blue with a stain that's staining the DNA. And then the telomeres are visualized with a fluorescent probe that recognizes the telomere. And the intensity of this spot, that is how many probes are hybridizing there, is proportional to how long the telomere is. So the brighter the spot, that means you have a longer telomere. And this is a very nice assay because you can actually see what's happening to the chromosomes. So this is how we were able to look at telomere length. And what we found when you breed these mice down in this nice line here is if you look at the intensity of each one of those red dots from before versus the number of dots that have that intensity, you have this nice distribution. And this distribution is the normal way that telomeres are maintained, and they're maintained about this equilibrium because there's some shortening and lengthening and shortening and lengthening. However, with each generation now, this is the G2, the G4, and the G6, you can see that there's progressively shorter and shorter telomeres with each generation that these mice are bred. One of the nice things about this visual assay, where you're actually looking at the chromosomes, is that we could show that not only are we losing telomeric sequence with each generation, but we're actually losing telomeric function. And I'll be telling you in just a minute that we see effects, that is, phenotypes in these later generation mice that we don't see in the first generation mice, indicating that the short telomeres are what's causing effects in these mice. So when we actually look at the different generations of mice, here is the wild type mouse chromosomes that I showed you with these red dots on the ends of the chromosomes. And you can see that these mouse chromosomes look like these little boomerangs. They look like little V's because the centromere is all the way along one end of the chromosome. So they're attached here, and then the two long arms are going off here. So you see this nice spread of mouse chromosomes. In the second generation, you also see that, and also the fourth, and in the sixth. But what was remarkable is that in these very later generations, what you see here is what looks like two chromosomes that have now fused through their ends. Instead of being little boomerangs, they're two that are attached to each other. And what that indicates is that not only have we lost telomere sequence, but we've lost telomere function. Because I told you that what telomeres do is to protect the chromosome ends, and it protects from these kinds of recombinations. So we can actually see this loss of telomere function visually. So the mice, once they get to be in the fifth and sixth generation, it turns out have a number of problems with them. So I mentioned in the testes that these mice become infertile. So there's actually cell death of the germ cells. In the intestine, the normal intestinal cells, which allow cell turnover, isn't occurring properly, so you have this atrophy. In the blood, there's bone marrow failure. So the blood is one cell type where you have to have cells continually renewing because blood cells are dying every day. In the skin, there's decreased wound healing. and there was premature graying of these late generation mice. And it was remarkable that all of these phenotypes seen in these mice were seen in the 5th and 6th generation mice. Okay? They were seen when the telomeres were short, but not in the 1st generation mice when telomerase was absent. So what that tells us is that it takes some time for the telomeres to get short, and it must be the short telomeres that are causing these effects. Okay, so it's not the absence of the telomerase enzyme, because if it were the absence of telomerase, you would see that in the very 1st generation. The fact that we have to wait for 6 generations means it must be the short telomeres that are causing these effects. So I'm going to tell you now about work from a student who was able to show that it was actually the shortest telomeres that caused this effect. And this was Mike Heeman. And Mike did a series of experiments crossing mice with long telomeres and mice with short telomeres. And what he was able to show is that it's actually the shortest telomere that leads to either cell death or senescence. So just like the different generations of mice, the mice with the long telomeres, there's no effect. As those cells are dividing now, the telomeres become short, and the shortest telomere in the cell is actually recognized, and it tells the cell that there is damage going on, and what happens is the cells either undergo cell death or cellular senescence. So cell death, you actually get the absence of the cells, and cellular senescence is a phenomenon where the cells just stop dividing and they won't divide any further, but they're still staying around. So it is clearly the short telomeres that are causing this effect. So we were interested then in the consequence of this short telomeres causing cell death. And so I'll tell you one short story where we were interested in how this may play a role in cancer. So we were interested in a particular kind of cancer, and that's a cancer that causes B-cell lymphoma. So this is a leukemia-type cancer. And a model mouse had been developed, and we call that mouse emu-mic because this expresses a particular oncogene. And this mouse had been characterized and is very tumor-prone and will develop this B-cell lymphoma. So what we were able to do then is to cross this with the telomerase null mouse and ask what happens when telomerase is not there. Can that tumor go on and develop? And so, of course, we have to be able to understand what happens when you first get rid of telomerase. So we look at the 1st generation, the emu-mic mice that are G1, and then, of course, have to breed and get the G2 mice, the G3 mice, et cetera, all the way up through the G6 mice to understand the role of the short telomeres in this development of the tumor. So what we find is that there's a change in the survival of the mice due to these tumors. And so I'll just walk you through this graph here. This shows the percent of mice that are alive at the beginning of the experiment versus the number of days. And so what's shown in the black line here is this mouse which has been made prone to these B-cell lymphomas. And so at the start, 100% of the mice are alive. But if you follow this black line, you can see that as time progresses, at 100 days, about half of these mice have died. And they've all died of this B-cell lymphoma. So they get this tumor at a very high rate. If we now look at the mice which were absent for telomerase, so they now don't have telomerase, but they're the very 1st generation that lack telomerase. What you find is a very similar curve here in blue. So all of these mice are dying of B-cell lymphoma, and they're all have succumbed to this lymphoma by about 200 days. However, in contrast, when we breed that out to the 6th generation, where now the telomeres are very short in these mice, what we find is that although there are some mice that die early on, most of the mice now survive. And what you can see in these mice is that a lymphoma started to grow, but then it stopped growing. And it stopped growing because the telomeres were very short. And as I told you, the short telomeres then cause either cell death or cellular senescence, so they couldn't go on and form that tumor cell. And so this is one indication that the short telomeres will limit the growth of cells, and in this setting, it's limiting the growth of these cancer cells. So the short telomeres will block the tumor growth. They don't block the early stages that cause the cancer, but what they do is they block the growth of the cancer cells. So the telomerase itself isn't playing a role in the initiation of the cancer. However, when you have short telomeres, after those cells start to grow for a while, you now block at this stage, and the cells can no longer go on and form the full-blown tumor. So this was the work of David Felder, who was another graduate student in the laboratory who was breeding these telomerase null mice to the B-cell lymphoma mice. So the other aspect I would like to focus on now for the rest of the talk is the role of telomerase in tissue renewal. So we were very interested in some of the early effects that we saw in our 6th generation telomerase knockout mice, and we wanted to understand a little bit more clearly what happens in normal tissues when you don't have telomerase and the telomeres become short. So a number of years earlier, we had been involved in a collaboration with Cal Harley, where we had shown, and others have also shown, that if you take normal human cells and you put those into culture and you grow them for a number of cell divisions, it turns out what happens is that the telomeres shorten progressively as the cells are dividing. So this is this end replication problem that I told you about. And so what is shown here is representation of the distribution of telomere lengths in human cells. Again, there's this heterogeneity of the telomere lengths. And as these cells are dividing, the length of this telomere actually decreases as the cells are dividing in culture. So this was a first indication that telomerase may play a role, that short telomeres may play a role in the inability of human cells to continue to divide. And then a similar phenomenon is actually seen in people, not in cells in culture, but rather with people of different age. So if you look at the mean telomere length, and the way that we get that mean telomere length is this is a representation of the telomere length. And so you can determine how long a telomere is here versus how long the mean telomere is here. And if you simply plot this on this graph here and look at mean telomere length versus the age of the person from which you took those blood cells, so this is now in normal human white blood cells, what you find is that there is a progressive shortening of the telomeres with increasing numbers of cell division. Now, at first, what we thought that this might represent is the fact that these white blood cells might not have telomerase activity, and therefore the telomeres are shortening progressively. Although what I'm going to show you is that they do have some telomerase activity, but the number of times white blood cells have to divide outstrips the ability of the telomerase to be able to keep up and elongate the telomeres. So over this long course of cell divisions, what you see is this progressive telomere shortening in the bulk of the white blood cells in all normal individuals. So the point that I'm going to make is that telomerase is limiting in cells. There's really just enough telomerase activity in order to keep up with telomeres on the chromosomes that are where the telomeres are the shortest. So this story comes from a number of different angles, but the first hint that the short telomeres might play a role in human disease came actually, again, from understanding molecular components. Just like understanding the molecular component of the telomere DNA sequence allowed us to understand what happens with telomere shortening. In this case, it's understanding the structure of the RNA component. So the telomerase RNA has this structure here. It was first identified with Kathy Collins' lab here at Berkeley, and also in my lab, that there's this particular structure here which binds to a set of proteins. In addition to this TERT protein, which is necessary for telomerase activity, there's a set of proteins that binds to this structure. And one of those proteins involves this protein here called discarin. And this discarin protein was known to be involved in human genetic disease. And so that then led to just the next year after this, a publication by a group of human geneticists who were very interested in this disease, dyskeratosis congenita. And I'll tell you just in a second what dyskeratosis congenita is. But what was happening here is that this group was very interested in this inherited human disease. And so they were mapping in families this disease, not knowing what caused it. And by doing this genetic mapping, they were then able to identify that in this case, the cause was the RNA component of telomerase. So the title of the paper that was published in 2001, the RNA component of telomerase is mutated in autosomal dominant dyskeratosis congenita. So this really galvanized people to understand how it is that telomerase can play a role in this inherited genetic disease. So the clinical manifestations of dyskeratosis congenita, it gets its name because of skin and nail problems, fingernails and toenails. That's why the term is dyskeratosis, because of the keratosis in the fingernails and the skin. So that's how the physicians would see their patients. That was the most common visual manifestation of this disease, although it was known that the mortality of the disease was due to bone marrow failure. Okay, so the inability of the bone marrow to continue making all of the normal blood cells that we require every single day. So this is an example of this requirement for stem cells to differentiate into all of the different blood cell types. So that was the initial characterization of the human disease. And the question was, how is it that the telomerase can play a role in this human disease? So we got involved with this at Johns Hopkins because a patient came into the hematology clinic at Johns Hopkins. And as I was mentioning, we don't do science in a vacuum. We talk to our colleagues. And I was talking with a variety of the people in the clinical departments to understand how to study the blood cell effects. And this patient came in, and this is the family of this patient, but this patient came into the clinic and was diagnosed as having this disease, dyskeratosis congenita. And so at the time, Mary Armanios decided to understand more about this family because what was known was that this keratosis congenita was often mutations in this RNA component of telomerase. So she brought these individuals within this family into the clinic and was able to identify that there weren't mutations in the RNA component of telomerase, but there were mutations in the protein component of telomerase. So again, this was a telomerase-mediated disease. And I show you this family, this individual that came into the clinic, and his father and his grandmother. And those individuals here shown in black had these changes, these mutations in the telomerase in the family. So this inheritance of this disease was shown to be autosomal dominant. And I'll tell you a little bit about what we know about that autosomal dominant inheritance. One of the remarkable things about this family was that the disease showed something that we call genetic anticipation. And that's a fancy way of saying that with each generation that has the disease, there's an earlier onset of the disease and more severity of the disease, okay? And this may remind you of our telomerase knockout mouse. In the telomerase knockout mouse, we didn't see anything in the first few generations, but then we saw effects in the later generations. And so the same thing seemed to be occurring in this human family with this inherited disease. The other thing that was really quite remarkable is that those people that were affected with the disease and had this bone marrow failure actually were heterozygous. That is, they contained one normal copy and one mutant copy of the telomerase gene. And that was quite remarkable to us because in a lot of human genetic diseases, you have to have two mutant copies in order to actually have an effect. So this really got us thinking about how could it be that you could be heterozygous, that is, have one normal copy and one mutant copy. And so as geneticists, what we thought about was two molecular mechanisms that might underlie this inheritance pattern in these families. And one of those is what's called dominant negative, and the other is haploinsufficiency. So what that basically means is that if you have one mutant copy and one normal copy, it could be that the mutant copy actually binds to the normal copy and takes it out of function. So that's a dominant mechanism, that the mutant is dominant over the normal copy. An alternative would be that you have one mutant copy and one normal copy, and it's just not enough to have one normal copy of the telomerase. And so we realized that we were in the position where we could test this genetically directly in our mice. So that's what we set out to do, was to determine whether there's a dominant effect or whether it's haploinsufficiency. And what I'll tell you is our evidence that it's actually haploinsufficiency. It's just not enough to have only one copy of the functional telomerase. So what's shown here is a representation of the telomeres again. This is a southern blot, but now we're looking at mice. So these are the mice that we had initially been characterizing. And you can see that there's this very, very long, heterogeneous size to the telomeres in this particular strain of mice. However, we looked at a variety of other strains of mice, and this is one that's shown here. And you can see that the long telomeres that are here in this initial strain now are represented by a much less heterogeneous and shorter telomere length in this other strain of mice that we call castaneous, for cast EI. And so we thought that we would be able to see what's actually happening to telomeres much more easily on this new genetic background. And I'll just represent that here. So if you imagine that you have this telomere length with the signal intensity of those red spots, the original mouse that we were using had very long, heterogeneous telomeres. However, this new mouse has much shorter, much more homogeneous telomeres. And so what we imagined might happen would be after you have telomere shortening, if there's a threshold below which the telomeres have to go before you have some sort of functional consequence, when you have telomere shortening in these settings, a much higher proportion of the cells would fall below this threshold when you have this much more homogeneous telomere length. And so that's what we had set out to do, was to generate mice that now have a much more homogeneous set of telomeres. And these experiments actually were initiated by Mike Heeman, who I showed you earlier, and then continued by Lingyang Hao, who then really had some remarkable discoveries. So what Lingyang set out to do was to take these mice now, where we can see phenotypes more easily, and to ask what happens if these mice are always have one normal copy and one mutant copy. That is, they are maintained as heterozygous mice. Okay, so rather than breeding these mice and looking at the mice that have no telomerase, now we're breeding these mice and looking, just like in the human studies, of always having the case of having one normal copy and one wild-type copy. So that's what we call the heterozygous generation 1, heterozygous generation 2, heterozygous generation 3. And what we saw was, remarkably, there is progressive telomere shortening as you breed these mice that have just one copy of telomerase instead of having two copies of telomerase. And so I'll just quickly show you that data to make that point. So if you have the signal intensity here, in wild type is shown in black, so you have this telomere length distribution. The knockout mouse have a shorter telomere length distribution, as we had previously shown. But what was really important is that the heterozygous generation 1 mice, shown in gray, are shorter than the wild type. And more importantly, the heterozygous generation 5 mice are shorter than the heterozygous generation 1, showing that we actually have progressive telomere shortening when you have haploinsufficiency. So this was quite remarkable. And that gets back to the point again that telomerase is limiting in cells, that having just half the level over a number of generations doesn't allow you to be able to maintain telomere length. If you now look at the survival of those mice, and this is once again one of those survival curves that I showed you earlier, with the percent of mice alive versus the number of days. And I'll just have you focus on 2 of these curves. The wild type, so in black up here is the wild type mice over 400 days. And down here, the heterozygous generation 10 mice. Now you see that there's a significant death of these mice due to the short telomeres. And there's actually, with each generation, there's a progressive decline in the viability of the mice. And this looks very much like the genetic anticipation that's seen in the human families that have dyskeratosis congenita. So the inheritance of short telomeres decreases survival. And as I said, this resembles the genetic anticipation seen in dyskeratosis congenita. So these telomerase deficient mice have many of the same defects that are seen in the human patients. Bone marrow failure, intestinal atrophy, immune senescence. They're chemotherapy intolerant, and they show this genetic anticipation. And the mortality in these mice is due primarily to these immune system defects. And all of these experiments would not be possible were it not for a really remarkable mouse handler here, Margaret Strong, who did all of the intricate breedings for these mice. So one of the most interesting things about this set of experiments was, of course, every time you breed 2 mice that are heterozygous, have 1 copy that's wild type and 1 mutant, you get out a heterozygous generation, and we can breed these for many generations. But at each stage of breeding, we get out, of course, the wild type, the next generation heterozygote, and we get out the knockout. And I can remember when Lingyong Hao came into my office one day, and he said, I wonder what would happen if we looked at the telomeres of the wild type mice that come from these crosses. And so he did that, and what I'll show you here is we designate that wild type star because it turns out that with the progressive breeding in the heterozygous state, even the wild type telomeres that come out were short because telomerase is limiting and the telomere lengths weren't restored. So that's shown here. Again, here's the intensity of the telomeres, the length of the telomeres. Here's the wild type telomere length. The knockout, just for reference. The heterozygous generation 5, I showed you earlier, is shorter than wild type, but quite remarkably, the wild type 5 star, that is the littermates, the brothers and sisters from these heterozygous mice, also have telomeres that are significantly shorter than their wild type counterparts. And that's because telomerase is limiting. You don't have enough telomerase to go around and top up all of the telomeres in just 1 generation. So I've been making the point to you that it's the short telomeres that are causing these phenotypes. When the telomeres get short, you cause a damage response and the cells die. So quite remarkably, when we looked in these mice, these wild type star mice also had the same effects as the mutant mice had. And this, as I said, is quite remarkable. So if we look at either the testes apoptosis, that is the cell death that occurs in the testes, or the immune system defects. If we look at the spleen weight, the wild type star mice had significant reductions. So this was really quite remarkable because now we have an inherited genetic defect, but even now in the case where the genetics is restored, the telomere length hasn't been restored and we still have an effect. So we called this this genetic disease in the absence of the telomerase mutation that caused the disease, we called that an occult genetic effect, a hidden genetic effect. And it's because the telomeres themselves haven't yet been restored to their normal telomere length. And now this might be a kind of a cute little trick that is, okay, so we have something, we now call it occult genetics, but it actually matters because, recall, this is an inherited human genetic disease, and you have these individuals walking around that have heterozygous mutations, and the only way to treat a number of these individuals, for instance, who have bone marrow failure, is with the bone marrow transplant. So you can imagine if you have a telomerase mutation in the family, and you have one brother that needs a bone marrow transplant because he has short telomeres, that his brother or his cousin, who may not have the mutation in telomerase, might still have short telomeres. And so it's important to know what the telomere length is of the donor bone marrow rather than just the genetics. So this actually matters to the treatment of this human disease. So it's the short telomeres, not the absence of telomerase, that's causing the disease. So just to round out this story, it turns out that having identified this genetic anticipation, where you see this worsening of phenotype in an earlier onset of this bone marrow failure, it turns out that there are other diseases that these short telomeres are causing besides the bone marrow failure. And so the first one to be identified was something called idiopathic pulmonary fibrosis. Idiopathic because we didn't know what caused it. And it turns out that it's caused by the short telomeres. And this was recognized by Mary Armanios because she saw this genetic anticipation in this family that had come into the clinic. And so it was identified that it was actually telomerase mutations that underlie this other human genetic disease. And as we've been able to understand more fully, Mary has been able to characterize what she calls the syndrome of telomere shortening. That is, there are a number of different effects of having these short telomeres. There are these bone marrow failure effects in the blood, the lung effects, the pulmonary fibrosis, liver disease, and as these studies are continuing ongoing, it's clear that there are going to be a number of different effects. And very interestingly, all of these diseases are actually diseases that are associated with the normal aging process. That is, they are normal age-related degenerative disease. So even in families that haven't necessarily inherited one of these mutations, if you have short telomeres, you may be at risk for a number of these age-related degenerative diseases due to the short telomeres. So this is taken from another group, and this is just an example of what I had shown you previously. If you look at telomere length versus age, and this is now just looking at white blood cells from normal people, what you see is that there is this decrease in telomere length with age, but also what's really quite remarkable is there's a huge amount of heterogeneity within the human population. If you look at a large number of individual people, you find that there's this great decrease. And there's been a little bit of information in the lay press about how maybe telomeres can tell you how old you are. However, due to this, of course, hopefully you would know how old you are, but the telomeres could determine if you didn't know how old someone was. But if you look at the heterogeneity here, and I gave you a sample of DNA, and that DNA had telomeres that were 7 kb, okay, that person could be 20, or that person could be 70. So it's not something that's so determinative that we can say for sure if you have a certain telomere length that there's a particular effect. However, this heterogeneity and this decrease in telomere length in the population, those individuals that have the shortest telomeres, we know are at risk for disease. So you can model this heterogeneity in the population with confidence intervals, and this was done by Peters Lansdorp, and Mary Armanios has also done that at Johns Hopkins. And so what you get here is this normal distribution. So if you look at telomere length versus age, these lines here were developed on 400 normal individuals, and you can say that if you're above this line, then that's the 99th percentile of telomere lengths. And so this is the normal distribution of telomere length in the population. And if you look now in a number of these families where you see this inherited genetic disease, those people in the families that do not have the telomerase mutation fall here around this 50th percentile. However, those individuals that do have the mutation typically fall below the first percentile. So those individuals with the shortest telomeres are the ones that are at risk for the bone marrow failure, the pulmonary fibrosis, and the liver complications. And as I hope that I've been making apparent, that since it's the short telomeres, not the mutation itself, that causes the disease, any individual that falls below the first percentile may be at risk for these disease, not just those that are in these families with these inherited syndromes. So many diseases that are seen in families with insufficient telomerase share features of these age-related degenerative disease. So we know that bone marrow failure is, in the normal population, an age-related complication, as is immune senescence, chemotherapy intolerance. Pulmonary fibrosis typically is diagnosed in the 6th or 7th decade. Liver disease, and also, paradoxically, there's this increased cancer incidence. So the short telomeres may play a role in the wider population, not simply in the subset of people with inherited mutations in telomerase. So just to summarize, I've told you that telomeres are required for chromosome end protection. Telomerase is essential for telomere length maintenance. Telomere shortening leads to either cell death or cell senescence after many cell divisions. Short telomeres limit tissue renewal and contribute to age-related degenerative disease. And then just to come back to the point about fundamental discoveries, new discoveries often come from unlikely places. When we set out to understand how it is that telomeres are maintained on the ends of chromosomes due to replication, we weren't looking to understand age-related degenerative disease or cancer. But by following our curiosity, it turns out that the telomere clearly plays a role in both of these diseases. So curiosity-driven research provides unexpected discoveries that can have important implications for human health. And these are the current members of my laboratory. We collaborate with Mary Armanios and her group, and many of the experiments that I told you were done by past members. And this is just a slightly older picture of my laboratory when they all dressed up as chromosomes. And then the laboratory currently, it's a little out of focus, but this was the political campaign for the American Society of Cell Biology. We are research giving a face to basic science. So I'll stop there.